friends, in today's lecture, we will talk about DNA repair mechanisms and pathways. DNA is the basic unit of inheritance and maintains the integrity and function of living organisms. However, it is constantly exposed to damaging agents which can cause DNA damage. These agents include radiations like X-rays and UV rays and many chemical agents like alkylating agents, hydrocarbons and antitumor agents. Additionally, errors can occur during DNA replication leading to harmful mutations. To overcome the harmful effects of DNA damage, cells have various DNA repair mechanisms which cope up with the damage and errors and repair the DNA. Although all cells possess a large number of different types of DNA repair mechanisms, each is relatively specific for a certain type of DNA damage. These repair systems can be grouped into two main categories: direct repair and excision repair pathways. Direct repair pathways include photoreactivation, repair by ADA protein, and repair by AlkB protein. Excision repair pathways include nucleotide excision repair pathway, base excision repair pathway, mismatch repair pathway, and the SOS response or SOS repair. In today's lecture, we will talk about direct DNA repair. And in the coming lectures, we will be talking about the excision repair pathways. Although it might seem that direct reversal of damage would be the simplest way to correct the damage, in most cases, the reverse reaction is not possible for thermodynamic or kinetic reasons. In a few cases, the reaction is reversible, and in some of these cases, mechanisms have been developed to take advantage of that reversibility. Still, there are very few examples of direct DNA repair. Albert Kellner was trying to measure the effect of temperature on repair of ultraviolet damage to DNA in the bacterium Streptomyces. However, he noticed that damage was repaired much faster in some bacterial spores than in others kept at the same temperature. Obviously, some other factor than temperature was operating. Finally, Kellner noticed that spores whose damage was repaired fastest were the ones kept most directly exposed to light from a laboratory window. When he performed control experiments with spores kept in the dark, he could detect no repair at all. Thus, he realized that light played a major factor in DNA repair. The light repair came to be known as photoreactivation and was the first DNA repair process to be discovered. Let us understand the details of this repair process. This is a DNA duplex. When exposed to UV radiations, it leads to pyrimidine dimer formation between two adjacent thymine moieties. We have already seen the effect of UV radiations in one of our previous modules. You can visit the channel website and look up for this video on physical agents or physical mutagens. It is catalyzed by an enzyme called Photolyase. This photoreactivation repair process is catalyzed by an enzyme called DNA Photolyase, and it is mainly responsible for the repair of these pyrimidine dimers in DNA which are caused due to the exposure to UV radiations. This enzyme can actually bind DNA even in the dark. It scans the DNA, and wherever it comes across a distortion due to pyrimidine dimer, it will stop there and it will bind to it. When exposed to light, it gets energized by the energy derived from the visible light and utilizes this energy of the photons to break the cyclobutyl ring structure between the two pyrimidine dimers. Thus, the damage is repaired and the photolyase is finally released. This enzyme photolyase is actually made of two subunits which are coded by the two genes PHRA and PHRB. Photolyase contains two chromophores responsible for absorbing the light energy or the energy of the photons. In all such photolyses, one of the chromophores is methylene tetrahydrofolate or 8-hydroxy-5-deazacflavin and the other is FADH. Photolyase can bind to DNA at any region and it then scans the DNA and stops wherever it comes across a distortion due to the pyrimidine dimers. The enzyme binds to the thymine dimer and the first chromophore folate is used to harvest the light energy. This energy is quickly transduced to FADH, making it FADH2. The now electronically excited FADH2 can transfer the high energy electron to the dimer, causing the cyclobutyl ring to break and separate the two thymine molecules again. However, the distribution of photolyse is not universal. Photolyses are found in bacteria, fungi, plants, and many vertebrates, but not in placental mammals. It is widely present in microorganisms, but it is absent in Bacillus subtilis. It is present in Saccharomyces cerevisiae, but absent in Saccharomyces pombe. In addition, 6,4 photolyses have been found in insects, reptiles, and amphibians, but not in E. coli, yeast, or mammals. So let us quickly revise this light repair mechanism or photoreactivation. As you can see, this is a DNA duplex. Due to the exposure to UV radiations, pyrimidine dimer or thymine dimerization has taken place between two adjacent thymines here. Remember, the ring formation takes place between the fifth and the sixth carbons of the thymines to form the cyclobutyl ring. This enzyme photolyase now scans the DNA, binds and scans the DNA, and when it comes across this thymine dimer, it halts here. When exposed to light, this photolyase is activated and it absorbs the light of the photons. And as we have already seen the mechanism, it is responsible for breaking this cyclobutyl ring between the thymine dimers. And when it has broken this ring between the thymine dimers, the enzyme is then released and the DNA damage is thus repaired. Let us now move on and talk about another direct DNA repair example, that is direct repair of alkylated guanine. If you remember from our module on chemical mutagens, the guanine is alkylated at O6 by alkylating agents like ethyl methane sulfonate and methyl methane sulfonate. An enzyme called O6 alkyl guanine DNA alkyl transferase is responsible for the repair of O6 alkyl guanine. This enzyme is also called ADA protein encoded by ADA gene in E. coli. It is made up of 354 amino acids and at the 321st position, a cysteine residue is present. The guanine in this DNA duplex has been methylated to form O6-methylguanine. DNA methyl transferase or ADA protein approaches the site. It binds to the damaged DNA site and transfers the alkyl group of the alkylated guanine to the sulfhydryl group of the 321st cysteine residue. Thus, the enzyme now becomes alkylated and the damage is repaired. But the protein loses its activity and hence this enzyme is not an enzyme in the correct or the strict sense. Unfortunately, once the alkyl group has been transferred to the protein is permanently inactivated because alkyl cysteine is very stable. Additional repair requires the synthesis of new protein molecules. To help accomplish this in E. coli and perhaps in other organisms, The alkylated version of the protein induces increased transcription of the gene encoding the protein. The alkyl groups handled by this protein range in size from methyl to benzyl. Homologs of these enzyme or protein are present in all living organisms. A third example of direct DNA repair is direct repair of 3-methyl cytosine. This is brought about by AlkB, a protein that is found in most, perhaps all, living organisms, including humans. This protein is a member of enzymes called alpha ketoglutarate dependent and iron dependent oxygenases, which use iron oxo intermediates to oxidize chemically inert compounds. In the process, alpha ketoglutarate is converted to succinate and carbon dioxide, which is similar to one of the steps in the tricarboxylic acid cycle. AlkB is capable of reversing methylation at position 1 of adenine and structurally similar position 3 of cytosine, as can be seen on your screen. The reaction catalyzed by AlkB couples the oxidative decarboxylation of alpha-ketoglutarate acid. This leads to the hydroxylation of the methyl group. The methyl group then spontaneously decomposes to formaldehyde, restoring cytosine. Thus, the original base is restored and the damage is repaired. A final example of direct damage reversal is the sealing of a subset of nicks in DNA by DNA ligases. However, DNA ligases can only seal nicks between 5' phosphates and 3' hydroxyls. Nicks with other configurations or nicks accompanied by additional backbone or base damage require more complicated processing prior to repair and would not be classified as direct damage reversal mechanisms. With this, we come to an end of this lecture where we have discussed mechanisms and pathways of direct DNA damage. In the coming lectures, we will be talking about the excision repair pathways like nucleotide excision repair pathway, base excision repair pathway, mismatch repair pathway, and SOS response. If you like the content of this channel, please stay tuned to this channel, like, share, and subscribe. Thank you.